looking forward to a fun afternoon talking to many of you. So actually, you know, I haven't done any research now for 25 years. But NIEHS, I got kind of a global view of the endocrine disruptor field and also the focus on windows of susceptibility. And that was those were my two major areas. So I'm trying to keep up with them, and I'll give you kind of my perspective, and most of it will be from animal studies, but because I'm here today, I put in one human study just so that make you all feel it fits in. Now, I should be able to figure out which one. That was the wrong one. I guess the push.
Hold on.
Just sort of, I should have just gone like.
Just that nose, yeah.
Okay, can you see with the lights on? Maybe we should turn the lights off in the room, huh?
That'd be better, yeah.
And some of these chemicals are toxic. Is this thing? It must have a.
A pointer, it just bounces off the screen, so you just have to gesture.
Oh, no, I'm going to make a mess.
Hold on.
Trying to figure out how that works.
Okay, you're good.
Okay.
These
But,
guys.
okay, I won't use the
No, the pointer doesn't work.
Just
pointer,
the arrow.
doesn't work. Okay. So some of these chemicals are perfectly safe, probably the majority. Unfortunately, we don't know that for sure because most of them aren't really actually being tested. You guys are the guinea pigs for testing of a lot of these chemicals. Some of them are toxic just because they kill cells. Some of them are toxic because they are mutagens. They interfere with DNA, they break up the DNA, and that causes cell death or things that are so terrible that the organism many times dies. But some of them are more insidious. They mimic hormones. So what's interesting is that these chemicals are designed for some specific purpose, like a pesticide or something. But just like when you're watching drugs on advertisements on TV, they got all these side effects that you hear. Well, these chemicals, designed for a specific purpose, many of them have side effects, and some of the side effects are that they interfere with hormones. They look like hormones, and so sometimes they act like hormones in the system. Sometimes they act like antagonists to hormones. And so they disrupt the hormone balance. And of course, they're everywhere. I like this slide. It's everywhere, every day, and everyone. We're all exposed. So it wouldn't, if we weren't exposed, then it wouldn't be a problem. But the CDC has tested over and found over 350 or so chemicals in human blood. There's a variety of chemicals that we find in 100% of people, a bunch of the phthalates and bisphenols. So there's a lot of exposure out there. And you can see some of them are herbicides, pesticides. A lot of personal care products have toxic chemicals in them. Some are surfactants, flame retardants, plastics, sunscreens, everywhere you look. So first we found there were endocrine disrupting chemicals, and then we're looking at the toxicity of these chemicals. And about that same time, about 30 years ago now actually, this idea that there was sensitive windows of susceptibility came about. And that actually started in humans and started with the focus on nutrition, but then got picked up quite quickly by people doing environmental chemicals. So everybody understands how finely tuned and orchestrated development is. And it's time sequenced, very carefully controlled by hormones and growth factors. So you can see, if you have an environmental chemical, or nutrition, stress, even infections, and some drugs, they can alter this developmental programming that the hormones are responsible for. And they do that by altering the epigenetic programming that the environmental chemicals are doing. And they then alter gene expression, they can alter proteins, alter numbers of cells or where cells are. But in actual fact, what's so insidious about this is these are really what we call functional changes. Everything looks okay. You don't see that there's any problem because the changes are within a tissue at the molecular level. And these changes then don't show up as increased susceptibility to disease until way later in life. So the chemical exposure can be at one time, many decades later, the effect shows up. So we call that a bad start lasts a lifetime. So why is it that these endocrine disrupting chemicals are so disruptive to the system? So on the right hand side you have a hormone, and this hormone works through these specific receptors, and that's all it does. They're all estrogen receptors of some type. There's more than one, but they're all just estrogen receptors. So here's BPA, which is found in plastic. And people say BPA is like an estrogen. So it can indeed bind to all of these different receptors just like estrogen. And if that were the case, that would be bad enough because it would be interfering with the normal hormone estrogen. But in fact, what happens here is these kind of chemicals don't have the specificity of a hormone for a receptor. So they bind to all kinds of different receptors. So you can see how they can really mix up things and cause trouble, not just by binding through one receptor, but all kinds of different receptors. So how do they do their bad deeds really here during development? Well, before I go there, let me show you this slide. This is my favorite slide because it shows a bunch of human diseases. So these are the most important human diseases today, really. And if you look at them, they're also the diseases that have increased the most in the last 40 or 50 years. And we can mimic every one of these diseases in an animal model with some of these endocrine disrupting chemicals. So that's really huge. So it's exposure during development, during pregnancy, and then later in life, we can see all of these different diseases showing up. And I just put a couple chemicals there just to show you that there actually are some chemicals that do that. Of course, we'd like to know these effects in humans. And in red are areas where we have strong epidemiologic evidence. And the reason we don't have it for all of these is because these are birth cohorts. And some of these diseases don't show up till age 20, 40, 60, 80. And none of these birth cohorts really are that old. Some of them now are 20 and 30 years, and we're starting to see some things beyond children. But there's a lot more to do there. So that really is the real bottom line statement that all these diseases can be caused by endocrine disruptors in animal models. And probably they're doing similar kinds of things in humans. So if we look at this field 10 years ago, the field was really focused on animal models and development in utero as the model. I've given literally tens of talks on this topic, focusing on, oh my goodness, development is the sensitive window. And NIEHS was focused on that. And all of the birth cohorts focused on that as the most important window. And then we get into molecular mechanisms. Everybody focused on methylation as the epigenetic change because that was the simplest one to measure. And of course, the fields were really separate. There were people studying chemicals in development, studying nutrition and development, and studying stress. But nobody was studying really the interaction. And the end result was neurodevelopmental, reproductive, and some cancers. So what's happened in the last 10 years, as everything has just exploded, as the young people say, this DOHAD paradigm has gone viral. It's everywhere. There are new sensitive windows of exposure. There's new diseases, new tissue, new chemicals, and expanded epigenetic mechanisms. And I'll go into some of these now to show you where we are. But I think this statement is really an important one for the whole field. So nothing in disease etiology makes sense except in the light of altered tissue programming. So you alter the tissue programming, that increases the sensitivity to diseases that show up later in life. So where are these chemicals having their major effects? And wherever it's been looked at seriously, and here are 3 cases, the effects are on the stem cells, which makes sense because the stem cells are developing and differentiating to form the different tissues. So in obesity, there's only 2 chemicals that have been looked at, tributyltin and bisphenol A, but the effect is on the mesenchymal stem cells. And what happens is these stem cells could become bone or fat, and when you have these chemicals there, they end up being fat at the expense of bone. Now if you look at, on the far right, the prostate, you see a similar kind of thing. You have normal stem cells, you expose them to chemicals, it reprograms some of their epigenetic mechanisms, and they're kind of pre-cancerous. But if you do nothing else, then you still are okay. But if you expose them to chemicals then later in life, that's the second hit, you end up with cancers and prostate cancer. And the middle one shows a nice example of uterine fibroids, chemicals in uterine fibroids. And this is my favorite example, and this work has gone on over 10 years and started very early. I think this is the best example of gene-environment interaction to cause disease. So what goes on here is you inject these animals with DES, which is a strong estrogen. You do it just for 5 days, right at birth. And then you sacrifice the animals a year and a half later and you look for tumors. So if you start out here, this is a normal rat. You take this normal rat and you give it DES for those first 5 days, you get no tumors at all. But if you take a rat that has a knockout of a tumor suppressor gene that's more sensitive to develop cancers, then if you look here, there are already 64% of them have these, well, it's not cancer, the uterine fibroids in this case. Then, if you give the DES on top of the genetic susceptibility and sensitivity, now you get 100% and they're bigger. So it's a really nice example of gene-environment interacting to cause the disease. Now what's the sensitive time window? You can give this DES early on, up through 5 days, and you still get this. You can give it 10 to 12 days and still get the same phenomenon, and that is because during that time, all the way through day 12, the tissues are still developing from the stem cells. Now if you go and you dose from 17 to 19, at that time you have already a fully differentiated, mature system, and then there's no effect. So you have gene-environment interaction and time. And what's going on? what's going on is this estrogen is sensitizing the system by changing gene expression of some of the genes. It doesn't change the estrogen receptors, but it changes the level of progesterone and androgen receptors. And it only does it in this tissue for fibroids and not in the liver, because at the time we're doing this, the liver is already fully formed. So what happens in the normal estrous cycle in these animals? Progesterone receptor goes up and down as it goes through the cycle, because the estrogen is going up and down and it's stimulating the progesterone receptor and the androgen receptor. But in the ones that were stimulated with DES, they're more sensitive to that estrogen, so these receptors are up all the time throughout the cycle. And you can see how having those receptors up at the wrong time would be disruptive to the tissue, and that then leads to increased susceptibility to the fibroids. And how does that work? The initial work from this lab focused on methylation, the endocrine disrupting chemical interacting with the receptor, and this is a non-genomic pathway through the PI3K and AKT pathway. And now what they're doing is focusing on, just like a lot of people in the field, on histone modifications. So activating this pathway activates some histones to open up genes and turn genes on, and it phosphorylates here and turns some genes off. And the genes that are turned on then, a lot of those are the ones that are responsive to estrogen. So that gives you a nice story from start to where we are now of one system and one chemical. But I think this is even more exciting, because what we always say is these chemicals interfere with development, and they produce tissues that are more sensitive to disease. So here's a really nice example. So what you just need to look at here, these are undifferentiated cells that when you stimulate them with rosiglitazone, they become fat cells. And you can see the difference in the genes here as they go from undifferentiated to fat cells. And it's the same situation over here. These are mesenchymal stem cells. They are controlled. Rosiglitazone turns on genes and they become fat cells. But if you stimulate them with tributyl, which makes fat cells, you can see they don't look like the rosiglitazone fat cells. They're making a fat cell, yes?
What is rosiglitazone?
Rosiglitazone is a, I can't think of, I think it's Avandia. It's a chemical used for diabetes, but it's a PPAR gamma activator actually. And because of that, that's a big pathway for causing the differentiation of fat cells. And so this chemical, in addition to being useful in diabetes, people who take it get fat. And it shows up here. They're making fat cells. So the fat cells that are formed by an endocrine disrupting chemical, in this case tributyl, they look like fat cells, they store fat, but they're incompletely differentiated fat cells. They have lower adiponectin. They don't downregulate their pro-inflammatory system. They don't have any Browning transcripts active. They have fewer mitochondria. So you can see there's more of them. They store fat, but they're abnormal, and they're going to be then more sensitive to inflammatory problems, which is one of the real side effects for obesity. Okay. So the question then is, which chemicals here will cause these dysfunctional adipocytes? And if you want to take that to the human situation, could we look at human adipocytes and look at their gene expression and see, are they normal or are they abnormal, and do they look like tributyltin, or if we had a list of a wide variety of chemicals here and show how they would modify fat cell development, maybe there would be biomarkers there that would give you an indication of which chemicals they were exposed to. Okay. So all of that is the windows of exposure that's developmental. When the tissue is forming, in utero and the first few years of life for some tissues, like for brain, it can go all the way up through teenage years and the brain is still developing. So a few years ago, people started asking other questions, which is the fun part of science. You're focused all these years on one aspect, and then some crazy person opens up this whole Pandora's box and says, well, maybe there's other windows of susceptibility. What would they be? And would they be through the same mechanism as here, which is altering epigenetics, or are there some other mechanisms? And then how would all of these interact to actually cause disease? So it's just a few years ago, this whole area now of preconception exposure. Instead of measuring exposure during pregnancy, you measure exposure a couple of months before pregnancy, in either the males or the females or both. And papers are just coming everywhere on chemicals and on nutrition, that the fathers really matter. It's not just during pregnancy, but pre-pregnancy fathers. So then another person got a bright idea. So if you're doing this developmental exposure, you have the dams during pregnancy, and then you follow the pups. And you don't do anything with the dams after pregnancy. So he said, well, why don't I look at the dams after pregnancy and see? His interest was in diabetes. He was doing bisphenol A, which caused diabetes in the pups. But what about the mothers? So he looked 3 months after, and nothing was going on. And by all rights, they should have just stopped the experiment and said, well, there's nothing going on here. But they kept the animals, and by the time they got to 6 and 7 months, they saw a huge effect. The pancreas, due to bisphenol A exposure in the mother, was falling apart. And beta cell mass was way down. And what was happening is less cell differentiation and a huge increase in apoptosis. So not only were the pups sensitized, but the mothers were sensitized. And that was the only paper in that field for 4 or 5 years. But the question is, how many chemicals would do that same kind of thing, to follow up the mother? And that's something epidemiologists could do very easily too. And maybe what kind of endpoints? Are all the endpoints going to be affected, or just certain ones? So which chemicals, which endpoints? There's a lot to do still there. But just last year, another person entered this field, still studying these BPA. This is BPS. It's an analog of BPA. So BPA is toxic, and so everybody wants to get rid of BPA. You have BPA-free everything now. So they replaced it with BPS. It turns out BPS is just as toxic as BPA, everything they've looked at. And she looked at the way the mothers were handling the pups. So in a normal situation, the pups are born, the mother huddles them in the corner, she's constantly watching over them. There's a lot of licking and grooming and nursing going on. But the ones that were exposed to the BPS, were actually bad mothers. Less licking and grooming. See, the pups are all over the place here. She doesn't really care about them at all. The nursing behavior is off. Her mammary glands are not producing the right amount of milk. So these poor pups now, you can see they have a double whammy of poor nursing and mothering, in addition to the fact that they were exposed to BPS during development and the different diseases, including obesity and stuff that come with that. Now this is another interesting area that developed, and this person, this is Jennifer Adibi. She's an actual epidemiologist. She's what I call the new generation of epidemiologists. Somebody who does human cohort studies, but also takes tissues and puts them in the lab and does molecular biology on the tissues. And she discovered something that I think she didn't even realize herself, that this was something so important. So phthalates, if you give them to the pregnant mother, they affect the fetal gonad, and one of the results is decreased testosterone produced in the gonad, and that changes your anogenital distance and a bunch of other endpoints, hypospadias. So that things that show up, the phenotype here, is because of low testosterone from the phthalates through the mother affecting the pups. Well, what she found out, she started studying the placenta. And what happens in this particular case is the phthalates affect the placenta. The placenta produces HCG, which should stimulate testosterone in the developing pups, but the placenta production of HCG is way down, and so it could be, while the end result is the same, the actual place where the toxicity is occurring is not in the phenylgonad per se, but the placenta production of HCG, which affects that. So that opens up then another whole field to take a look at. And it spurred a lot of interest now in looking at the placenta and endocrine disruptors that affect the placenta. The biggest and most disturbing change was to start looking at effects that occur in future generations. So what we've been talking about is exposure to this mother affects this pup, which grows up here, and then we've been analyzing this pup for diseases. But if you then mate this male, or mate this female down the line, the germ cells here make the F2, but then when you go to the F3 generation has never seen any chemical at all. So any effects have to be transferred through the germline, which everybody said was absolutely impossible because there were no epigenetic marks that would be transferred. They are all erased and put back on normally, and this couldn't occur. But some people started to look at this anyway. And you can also look at it if you just start by dosing the males themselves, then what you call transgenerational with no exposure can occur earlier in what you call the F2 generation. So now, probably about 10 years later, after probably the first 4 or 5 years, people not believing there could be such a thing, now it's obvious that there are transgenerational epigenetic inheritance. And if you look at it in human terms, it would be exposing the mother to the chemical. The effect shows up in the children and also the grandchildren and the great-grandchildren. And so far, this is the list of chemicals, including some nutrition, that will cause transgenerational effects. And the ones in yellow will cause obesity transgenerationally in the third and fourth generation. So this is really, really disturbing, because you have the exposure here, and the effects don't not only show up in the first generation, but they show up the next generation, the next generation, the next generation. So the disease incidence continues over many generations from exposure in the one generation. So for epi studies, some of the effects we're seeing now could have been exposure to the grandmother. It's not just the exposure in the mother today affecting her children, but some of that could have come from many generations before. Yes?
How many generations before it disappears? Do we know?
We don't know. Most people have never taken it out past 4 or 5. In rodents, if you do it in a C. elegans, they've gone 18, 19 generations and it stays. But in rodent models, nobody takes it any longer than this, because the idea was just to show there is an effect. But you're right, now we have to ask that question. How long does it stay? But there's something else that's even in this same paradigm of exposing the mother and looking at the offspring. Something else had showed up just this year, and unpublished, was just at the Endocrine Society. And what they're looking at just is vaginal opening, estrocyclicity, and folliculogenesis and GnRH secretion over generations. But what they did that was different, instead of using a single chemical, they used a mixture of androgenic and anti-androgenic chemicals. This was a mixture that was set up and studied by some other people, and they just took that and they put it at the lowest concentration with these 13 different chemicals and ran them over the generations. And something that had never showed up before really came out. So this is estrocyclicity in the offspring, and there's nothing there. But the estrocyclicity is really messed up in the third generation, which is the transgenerational one. So how do you get that? It doesn't show up in the first, but shows up transgenerationally. And not just the estrocyclicity here, but the changes in follicles with fewer antral follicles and a lot more of the atretic follicles. So there's some nice endpoints there. And then this one is even more striking, and these chemicals didn't have any effect on vaginal opening or puberty in the first generation, but look what they did in the second and third generation. There's a huge shift in onset of puberty, and that corresponds when they looked at GnRH secretion in the hypothalamus that was modified, that corresponded to the shift. So this is something now we got to pay attention to, that even the way we do experiments and looking at the F1 generation, if we see nothing, doesn't necessarily mean there's nothing there, because it's now showing up in the future generations. Science is like that, you know, things, you answer one question and it opens up 10 more, and that's the fun part of it, really. But here's what you should be worried about, because this is really the human situation. You have exposure to endocrine disruptors during pregnancy, and then you're born. Then throughout your lifetime, you're exposed to endocrine disruptors. And then you get pregnant, then you get pregnant, and you're exposed, and then you have children, and they're exposed, and they get pregnant, and they're exposed. This is the real human situation. And if this was the case, disease incidence could be compounded over generations. And look what that would do to the sustainability of the human race if the major effect that you're compounding here would be infertility, for example. So, any data to support this? One paper came out last year. And what they did is they took adult animals, I believe these are mice, and they gave them a chemical. In this case, they gave them estrogen. Instead of an endocrine disruptor, they just gave them an estrogen. And then they made it, and then this would have been the normal situation. You give the chemical, and then you go to the next generation and the next generation. But what they did is then they gave the chemical in the second generation and in the third generation or not. So they had a bunch of different possibilities. And that goes to back what I just showed you, giving it in each generation. And what they found is looking at fertility, nothing happened in the first generation. If you only gave it in the first generation, there was nothing. If you gave it in the first and second generation, there was nothing. But if you gave it in the first and third, you had these completely atretic testes and abnormal testis epididymis situations up there, an actual birth defect in addition to reduced spermatogenesis. And then if you had it in all three generations, the effect is more. So that's the only one experiment, so there's a lot to do, but that shows a really scary kind of paradigm, and it hits the worst one, fertility. And people talking all the time of how fertility is going down and down and down, been going down for the last 50 years or more. So moving forward, what do we have to do now that we know it's more complicated? There's all these other different exposure paradigms that we need to address. Not just in utero, but in utero, and then look at the mothers, or in utero and neonatal, and then adult exposures. We used to think there's nothing really going on there, but now we're thinking, well, maybe there's something going on there too, and it's adding to what's going on in utero. So you can do in utero, neonatal plus adult, and there's all these different preconceptions, and then the transgenerational. So it opens up this whole field. How many windows are there, and how do they interact with each other? We still believe that the in utero and in utero neonatal is the most sensitive, but these other ones could play a very important role too. So this whole field now is about to explode in all those different directions. What else is going on? There are new chemicals being studied. There's a thing called the Matthew principle, which says the more you study a chemical, the more people study that chemical. And the best example of that is BPA. It got so everybody was doing BPA, hundreds of papers a year, 300-400 a year in animal models. Got to the point NIEHS said, we are not going to do any more BPA. We got to move this field onto some other chemicals. So people are starting to move, and they're moving into flame retardants. Now they're looking at some pesticides, glyphosate, which is Roundup. You're starting to see human and animal studies showing toxicity of Roundup. They're taking another look at fluoride, acrylamide, which comes from barbecuing meats. And what are we doing to ourselves now with this marijuana? The question is, are we doing the same thing we just did with smoking? It took us 20, 30, 40 years to finally say, oh my God, that was a terrible thing. And now this is just going crazy without really any data to support that that's all okay. There are new endocrine disrupting chemicals coming out, and of course this whole idea of mixtures. And people are afraid of mixtures. You know, what mixtures should we use? How do we decide what's a good mixture? So there have been some funded studies from the European Union, and they came up with an idea that they're testing, is they're going in humans, measuring an endpoint, and seeing which chemicals affect that endpoint in humans. And then they're taking those chemicals at that kind of concentration and making a mixture and testing it in animals and seeing, indeed, are those chemicals causing that disease that would be predicted because they showed up in the humans. The other thing that's happening is the new BPA are perfluorinated. In the last 4 or 5 years, this whole field has gone crazy, and this is going to go for a long time because there's over 3,000 compounds in these perfluorinated chemicals. They're everywhere in cosmetics and food containers, and they're in military installations, and all this foam that they're putting out there, firefighters that have a lot of this kind of stuff in it. And one of the things that got us to reach such interest is what happened in North Carolina just a year or so ago, where they started measuring these perfluorinated chemicals in the water. And there's a specific river here. And so if you measure the chemicals up here, you get, that's A, you get some chemicals in there, but they're the old chemicals that you would expect because they've been produced for 50 years or more. And then down here, you have hardly anything. So something is putting these chemicals in up here. We don't know what it is, because this is what it really should be. But if you go down here to C, oh my goodness, there's some chemical has appeared that never was there before. And it turns out here's a chemical manufacturing company that's dumping this chemical called Genex in the lake, in the river. And this is a river that now people are drinking the water out of. And so there's a lot of concern for the dumping of this chemical in the river and exposure. Because these kind of chemicals are shown to have a lot of effects. Some of them, even in humans, they have effects on thyroid and obesity. So here's my human slide. And I really like this. This came out a year ago. And these people are looking at chemicals and in obesity. So there was an obesity study, and it was called the Pound's Law study. And the purpose of this study was to look at 4 different kinds of diets. High fat, low fat, this and that. And they followed them over 2 years. And what happened is people lost weight on all 4 diets, and basically the same amount of weight. And the first 6 months worked really well, and then what happened is they all kind of turned around. Even though they're on the diet, the weight kind of came back, and we all know that story, right? That's the typical human situation. It's so hard to lose it and comes back easy. Well, what they then went in and measured these perfluorinated chemicals, and it turned out that the women who gained the weight back the fastest had the highest levels of the perfluorinated chemicals in their blood. And then they went to see what that could be responsible for, and they found out that the ones that had the greatest weight gain and associated with perfluorinated chemicals had the lowest metabolic rate. So the perfluorinated chemicals were lowering your metabolic rate, so you weren't burning calories so much, which would lead you then to put the weight back on. So I thought this is really a novel, new type of thinking, taking a study that was already out there and measuring some other novel endpoints. We're also looking at different tissues and diseases, muscle, kidney. We're hardly doing anything in kidney. People aren't looking much at puberty, starting to look at hearing. more in neurodegenerative. We haven't done anything in psychiatric diseases and endocrine disruptors. NIEHS is going to start looking at that. And this whole immune diseases and stuff, all this stuff of gluten sensitivity and Crohn's disease and all this immune stuff, it just seems like somebody should be looking at that for endocrine disrupting chemicals affecting the immune system. Because we know there are a lot of them that do affect the immune system, and maybe some of that, since that's only come up in the last 10 years or so, what could be responsible for that? So, okay. But it's not all bad news. There's some good news, and that is that if we know the windows of exposure and we know the chemicals, we can do something about it. And so there are all kinds of things. You can reduce the production, reduce exposure, and if you are exposed, there are pathways that you can modify to reduce the effect, even though the chemical is there. And I can see I'm running out of time, so I'm going to speed up. I want to end with some thoughts on the field in general. Once I retired, and I had more time to sit around and think, and finally it hit me, when we watch the grants come in the endocrine disruptor field, I always called it throwing mud on the wall. Somebody did something here, and somebody did something there, and somebody did that. There's no coordination, it seemed like. All the endocrine disruptor people were working on their own. And when you look at that, there's no society to coordinate the field, do all the things societies do, have the committees, the fundraising, the help with funding, to help people work together and set up meetings and all that. There's also no degree in endocrine disruption, and you get no focus on mentoring. The whole field was just kind of a mess. And so it seemed like maybe something could be done. And there wasn't a lot of interest when I polled people in developing a new society, but they wanted something. So we developed this new website. It just came online about 2-3 weeks ago. It's called HEEDS, Healthy Environment and Endocrine Disruptor Strategies. So it's a nonprofit, multidisciplinary coalition of science dedicated to improving communication, coordination, and collaboration in the endocrine disruptor field. There's no membership fees or anything, but we're trying to pull people together through this website. So on the one hand, it's a website to go and learn a lot of stuff, and this is what it looks like. It's a typical kind of website, and we've got some highlighted parts here, and I'll go through them quickly in just a second. I think I can skip that. So here's what we're trying to do, trying to help organize the field a little bit. One of the things would be regional working groups. So I started a working group, a little kind of society in North Carolina. It's called EDC-NC. We have about 100 members. This last 2 weeks ago, we had our second annual meeting at NIEHS. We had 170 people sign up for the meeting, 36 posters, and the meeting didn't cost us a penny. All local people coming together, interacting, talking to each other. Now I'm going to try to expand that more across the country. And we're going to develop other groups. I've got a metabolism disruptor group that's got now about 106 people from all around the world that we share publications and ideas for sessions at meetings, and probably we're going to start developing webinars and things. And we want to do some more meetings. I think we need to have maybe another endocrine disruptor society kind of meeting, and publications and blogs and webinars, trying to pull the field together a little bit. So we have a newsletter, you can sign up for the newsletter. And we're developing a directory. You can fill out a bunch of information about yourself. So if you want to know who's doing endocrine disruptor work on perfluorinated in Sweden, we'll be able to tell you that. So there's a lot of stuff. There's over 25 pages on here. All the recent books, key publications, all videos, organizations. We're starting to develop educational materials. People who aren't teaching a class anymore can send us the slides for that class, put it on, it could help somebody else who's developing a new class. All that kind of stuff. So we have really big ideas, but we don't have any money, but it's up and running. And we hope that you will look at it. It's just heeds.org. Sign up for the newsletter, the consortium. If you got any interesting information about meetings that are going on, things like that, tell us about it, join the working group, and you can follow us on Twitter. We're having just a ball putting stuff out on Twitter. Of course, I don't know anything about that, but I have a colleague, Sarah Howard, who's doing all that stuff, and she loves it. So there you are. Thank you very much.